Yeah, as said by Philip in the introduction, my research field is on the placebo and nocebo effects in the motor domain. And of course, this is let's say, a topic that has a wide range of potential application and implications, going from pathology up to sport excellence. So I anticipate that my main interest in my lab is the placebo effect on basic motor functions, and I will give a definition of this later. But before going into this specific topic, just a brief mention of the fact that the placebo effect um, has an important influence in some movement disorders, and I would like to mention the knowledge we have up to now. And also, I would like to briefly mention what we know about sport performance. Then I will concentrate mainly on what we are doing or what we have done in our lab to tackle this effect from a behavioral, subjective, and neural point of view in different types of motor functions. And then, if I have time, I will present some preliminary results from an ongoing EEG study. I don't give a definition of placebo and nocebo effects because I think in this case we know, and we also had before a nice introduction on this. So, what do we know about the placebo effects on motor deficits? Well, here we have, let's say, a kind of huge literature, we can say up to now, saying that these effects work in many clinical conditions. In the last edition of the Handbook of Clinical Neurology, there is a volume specifically dedicated on the placebo effects in neurology. And we also had the opportunity to write a chapter on focusing on the placebo effect on motor disorders. And from this, let's say, review that we have written, it comes out that most of the knowledge we have now in the motor disorder, movement disorders, is related to patients with Parkinson's disease. So we have evidence coming from different assessment methods, like, for instance, the use of clinical scales or objective tools like sensor or accelerometers or subjective reports, showing that patients with Parkinson's disease have benefits from placebo procedures that are present not only on the motor symptoms like bradykinesia or tremor or rigidity, but also at a subjective level in the quality of life and also in some affective symptoms like depression. And some evidence has shown that these effects are related to changes occurring in the brain of these patients, mainly related to dopamine release in the striatum and in changes in the firing rate of the subthalamic nucleus. And this evidence comes from the lab of Fabrizio Benedetti and also De La Fuente Fernandez. So basically, we have neurobiological, neurophysiological changes occurring in patients' brain that explain the behavioral and the subjective benefits reported by the patients. In contrast to Parkinson's disease, knowledge on the benefit of placebo in other movement disorders is very limited. There are very few studies on dystonia or tremor, and we should also notice that these studies are mainly based on clinical scales or subjective reports. So we don't have up to now objective measures of an improvement. And also, this evidence is mainly derived from randomized control studies, trials, run to prove the effects of a real drug compared to the placebo arm. So basically, this study tells us that there is a placebo response in these patients, which is not the same saying that there is a placebo effect, because maybe, as you know, the placebo response can occur also because of other confounding factors, like, for instance, the regression to the mean. Instead, the placebo effect is due to a pure modulation, manipulation of the individual expectation or learning. So this should be taken with caution, of course, but having a placebo response in certain and a percentage of patients is a promising, let's say, indication of the potential also of a placebo effect if we carry out ad hoc studies. And then I would like just to mention another special condition, to my opinion, that is the case of functional motor disorders. This is a condition in which patients can manifest some motor deficits, motor symptoms like tremor or dystonic postures, which resemble those presented by patients with neurological disorders, but these symptoms are modulated by distraction. So they are subjected to distractibility. They disappear when the patient is distracted, and they are also inconsistent. So basically, these patients manifest motor disorders that are not explained by a neurological cause. Or let's say they are not explained by structural damage on the brain. In this case, placebo and nocebo effects are sometimes used by the clinician for diagnostic and therapeutic purposes. Indeed, these patients appear to have some nice placebo response, let's say. However, in recent years, also because of a new model to explain this disorder, like the predictive coding account, we propose that maybe the link between functional motor disorders and functional neurological disorder in general and placebo and nocebo effects could be deeper than previously thought. Our hypothesis is that maybe some nocebo-like mechanisms could explain actually the symptoms presented by these patients. So we developed this as a theoretical framework, a theoretical idea, but now we are running some studies to prove empirically these ideas. Going to sport performance, also in this case, we have a lot of evidence showing that placebo effects improve motor outcome for different sport disciplines. There are a lot of studies conducted by the group of Chris Beatty and Philip Hurst on runners or cyclists and also on weightlifters. And we have evidence, behavioral evidence, that athletes can improve their strength or sprint time or speed after placebo procedures. So here the question is now, what are the underlying neurobiological mechanisms that support these behavioral results? So now let's go to the main focus of my talk, which is the placebo effect on some motor function that we can also have in our daily life, like functions that allow us to have an optimal motor control when we interact with our object or with our surroundings. And I would like to present some work done with different types of motor functions from a behavioral, subjective, and brain level. With regards to the behavioral, and this is the beauty, I think, to study the placebo effect in the motor domain, because we can have an objective proof of a change in behavior after the placebo intervention. And we can eventually also ask participants some subjective questions in order to see whether the change in the behavior that we observe is matched by a change in the perception of the individuals or not. So, with regards to the behavior, the first question we have to ask is which body district we want to tackle, we want to study, whether we are interested in investigating fine-tuned motor control or very localized body district, or whether, like in sport performance, we are more interested in a general change in the volar functions of the body. And then, with regards to the body district, we also can ask ourselves which type of motor functions are we going to tackle? Are we interested in force or in speed or in accuracy? And because, of course, when we talk about motor performance, we are facing a multifaceted definition. So, with regards to fine-tuned motor control of very limited and restricted and localized body districts like the hand or the fingers of the hand or the upper limb, we have evidence with regards to force production. And these can be studied in different ways. Like, for instance, in our case, more than 10 years ago, we developed a task in which we were interested in recording the force produced by the participants with very basic a very limited movement of the finger, like abduction movement of the index finger against a piston connected to a force cell. So we could limit basically the muscle involvement to a very specific and localized muscle of the hand, which is the first dorsal interosseous, the muscle controlling this lateral movement of the index finger. Other groups are interested in more, let's say, a function of the volar hand, like in this case, the group of Piedimonte and Carlino in Turin. They use a grip handle to let the participant allow a force pressure with the older hand. And what we know from these tasks is that the placebo procedure can induce enhancement of force production. And this is consistent in many types of studies because we have replicated this finding in many studies. And I have to say here that the placebo device that allowed for this improvement in force production was a tangible device, an electrical stimulation, a sham electrical stimulation applied to the body part performing the task, because it appears from our studies that what is inducing a beneficial effect on motor performance is the fact that the placebo tool should be visible, tangible, and applied to the body district that we want to improve. Then we also have evidence that placebo effect can also improve movement precision. And we demonstrated this with another type of task, like a reaching task, in which participants are required to move the upper limb as quickly and accurately as possible from a starting point shown on a touch screen to a target, toward a target. And in this case, we use a classical Fitts law paradigm in which we change the task difficulty by changing the position of the target and the size of the target. Because we know that the difficulty of the task increases when the target to be reached is farther and smaller compared to when the target is close and bigger. And as you can see from this data, the index of difficulty, which is represented by these numbers on the x-axis, is linearly associated to the movement time. So basically, participants require more time when the task is more difficult. And this is the typical Fitts law, which explains the speed accuracy trade-off for goal-directed movement. The interesting thing is that this law is preserved after placebo procedure, as you can see by the red line, but the movement time is shorter, meaning that participant can perform the task more accurately and faster after the placebo procedure compared to a control condition. And in this case, the placebo was again an electrical stimulation applied on the upper limb with information that it was suitable to allow to improve speed and accuracy. And the strength of this study is that it was a within subject design in which the same participant was submitted to the control and to the placebo condition in different days. So basically, the same participant was the control of his own. Then we move also to a more sophisticated function, which is visual motor learning, in which we wanted to see whether placebo procedure are able also to induce an improvement in the ability to learn specific sequences of finger movement. And we use standard task to tackle this learning effect, which is the serial reaction time task, in which participants are shown some cues on the PC monitor. And these cues are these color visual stimuli that occur in different positions. And the task is to press as fast as possible a button in a keyboard with different fingers corresponding to the position of the visual cue. Unbeknown to the participants, in some blocks, these visual cues appear with a random order of presentation. Instead, in other blocks, they appear with a surreptitious sequence of positions that allows participants to enter in an implicit learning mode that we can see then in faster finger movements. And this implicit learning does not become aware because participants don't know that there was a sequence. And this occurs because the sequence is embedded in blocks of random trials. And what we can see then in the results is that there was an improvement in this type of task, so a better performance represented by a score in which we combine reaction time and percentage of error. There is an improvement after 24 hours from the training in the consolidation phase in the group of participants who received a placebo device, the electrical device applied to the end, with information that it could help in performing the task faster. Compared to a control group who was a natural history group because they did not receive any kind of intervention. We did not find the same benefit, significant benefit, in a placebo group who was submitted to another type of placebo intervention, which was transcranial direct current stimulation applied to the frontal area in a sham modus. So basically, it was not active, but participants of this group were informed that this intervention would have helped to improve concentration. As you can see, there is an improvement, but this was not statistically significant compared to the control group. And this suggests us that maybe when we work in the motor domain, maybe we need a placebo tool, placebo treatment that is focused on the motor functions. What about gross motor control of the whole body or of the lower limbs? In this case, there is evidence on force production with regards, for instance, to leg extension task from our group and also for other group. And in this case, since the task of exerting a certain amount of force is very challenging for the participants, what we observe typically is that placebo groups are stronger than control groups after the placebo procedure, but because they are able to maintain a certain level of performance throughout the procedure, not necessarily because they improve their performance at the end. We also tackle balance control. In this case, it was a task in which participants were required to control the balance by standing on the dominant leg. So it was like a single stance task. And we found that the body sways after the placebo procedure at T1 and T2 were reduced in the placebo group compared to a control group. In this case, the placebo intervention was again electrical stimulation of the leg. Finally, we also tackle gait by asking participants, these were older adults. Instead, in the previous studies, I was always referring to healthy young controls, so healthy volunteers, sorry. In this case, we tackle gait in older adults. They were individuals with more than 60 years old age, and we asked them to perform a gait task, a walking task on a sensorized carpet in two conditions. In one case, they were performing just a single task, so just walking at their comfortable, let's say, pace. And in another case, they were asked to perform the same walking task, but together with a cognitive task in which they were required to do some subtractions mentally. Typically, when we perform this dual task, we assist to a cognitive motor interference that basically introduce a cost in the gait parameters. So if we record gait speed or stride length, we can see that these parameters are worse due to the cognitive interference. But interestingly, we observed that the cost of the dual task was reduced in the placebo group after the placebo procedure compared to the control group. And in this case, the placebo device was TDCS, so the transcranial device, the direct current stimulation of the frontal area of the head, with the information that it would have helped to perform the cognitive task with less demand, with less cognitive demand. So, to our opinion here is that we can also leverage the placebo effect to let participants a better balance, to achieve a better balance of cognitive resources during the simultaneous execution of multiple tasks. We can also monitor if all these changes documented at the behavioral level are also present at the subjective level by simply asking a participant to rate their or to judge their performance or how they felt during the task. For instance, what we regularly monitor is the perception of performance with the visual analog scale. We ask participants how strong they felt, for instance, during the task, from 0, very weak, to 10, very strong. And we assist that typically, at least with the movement of the upper limb and of the hand, the subjective perception goes hand by hand to the behavioral outcome. So participants also feel stronger after the placebo procedure. This is also true for other type of perception. They also feel more precise when the movement required to be fast and accurate. They also perceive more stable in the case of the balance study. Instead, when it comes for, let's say, movement performed with a larger group of muscle, bigger group of muscle, like those of the lower limb, this subjective perception is not always in line with the objective performance, because in this case, we did not find any modulation in the subjective perception of performance, although our participants were better in the placebo groups than the control groups. Another type of subjective function that appear to be very interesting when working with the motor in the motor domain is the perception of fatigue. I don't mean here fatigability, so I'm not referring to the, let's say, amount of resistance to fatigue that we can manipulate during our procedure, but I'm referring to the perception of fatigue as a state, as a feeling that participant can have during the task performance. And what we can see, this is not very much studied, I must say, up to now. We only have up to now up two studies in which we investigated this. And we can find that participants in the placebo groups are not only better from a behavioral point of view, but they also feel less fatigue, both in a mental and physical level. And another function that appears to be very interesting for several reasons is self-efficacy. So basically, how much confident participants feel in performing the task after the placebo procedure compared to before. This is a very important cognitive functions because self-efficacy appears to be linked to the motor outcome in general. So participants who feel more confident to perform a task are also better in performing the task. So, and we saw that the placebo procedure can help to boost self-efficacy. Now, the question is, does self-efficacy, or let's say, does the placebo procedure improve the motor outcome because it works on self-efficacy? Or does self-efficacy increase because participants realize they were better in the motor performance due to the placebo? So basically, we can answer this question now thanks to some mediational analysis in which we want to prove the link between these variables by introducing self-efficacy as one of the main cognitive mechanisms at the basis of the link between placebo effect and motor performance. Now, as you know from the field of pain, the main mechanisms recognized as inducing placebo effects are expectations and also belief in treatment efficacy. And we constantly monitor these in our studies. Indeed, our participants also constantly expect to perform better and also believe in the efficacy of the treatment after having performed the task. But up to now, I must say, I never found a strong correlation between these two variables and the behavioral response. Instead, with regards to self-efficacy, we have promising results saying that maybe this could be a cognitive function that we should monitor and we should take into play when considering the placebo effect in the motor domain. Another aspect that is not very much investigated in the literature, but we know it can influence motor performance, is the focus of attention. And we also know from very few studies that the focus of attention, whether it is directed to the body or to the outside the body, can also influence the placebo effect. Indeed, in a previous study, we found that the placebo group induced to monitor their movement during the procedure, they also display a better placebo effect from a behavioral point of view compared to the control groups and to an external focus of attention group. But, here the evidence is still limited, although attention could be a function again that should be taken into account in this interplay between placebo effect and motor performance. And then there is also we should also consider some personality traits and states, like for instance, anxiety and optimisms. We know that anxiety plays a role in sport performance. And it could play a role also in the basic motor functions we are interested in, but we don't have a proof yet on the mediating role of these personality traits in this in the placebo effect on motor functions. What about knowledge on the brain level? So we know very clearly that the motor system is involved in all this, of course, because it's involved in motor control. So we know that the placebo effect can change the functions of the motor system in very specific way, because we have proof that the motor system is involved not only during movement execution after a placebo procedure, but also before movement initiation. For instance, by using transcranial magnetic stimulation, we have demonstrated that there is an increase in the corticospinal excitability before movement onset in participants who were expecting in the beneficial influence of the placebo on movement speed, for instance. And we also know that this brain modulation is very specific for the body district involved in the task, so not for other body district or for other muscle, and also for those body districts on which we have applied the treatment. As I said before, it appears to be a very localized effect. So, of course, we have to consider that motor control is not only related to the motor system, or let's say the corticospinal tract, but it involves a broader range or number of brain regions, both subcortical and cortical. And we have evidence of the involvement of subcortical regions, as I said at the beginning, mainly from studies in Parkinson's disease. But we also know about the effect of cortical regions in studies in healthy volunteers, both with transcranial magnetic stimulation, transcranial direct current stimulation, and also electroencephalography. And this neurophysiological evidence suggests that there might be many brain regions involved in the placebo effect in motor performance. And we are running now, I don't know, I hope I still have some minutes left to present some preliminary results, as I said at the beginning, on an EEG study we are conducting to enlarge a little bit the investigation of the brain regions involved from the motor system to other potential mechanisms. And for this, we use an EEG approach with a very basic task, which is a unimanual reaction time model task, in which we ask our participant to move the thumb as quickly as possible after the appearance of a go cue on a PC monitor. And we do this by let them receiving the go cue. As you can see here, the S2, stimulus 2 was the go cue inducing the movement. But this go cue was anticipated by a warning cue that appeared 3 seconds and a half before the go cue and informed participant on the forthcoming occurrence of the go cue. We let participants perform this task and we recorded the EEG throughout. We monitored the EEG activity and the motor performance before and after a placebo intervention, consisting of always the same placebo device that we apply in all of our studies, which was the electrical stimulation applied on the upper limb, the right upper limb, in particular on the forearm. And we had 3 groups of participants who received different informations about the effect of this TENS device. The placebo group was told that TENS was able to speed up movement. Instead, we had a control group in which we applied the same device by saying that they belonged to a control group, and so the device was applied at frequency that were inactive to change motor performance. And then we had a natural history control group in which we didn't have any kind of intervention. They just performed the task before and after. The placebo device was applied at frequency 10 hertz of frequency, which is completely inefficient to change performance. And what we can see from this preliminary result is that the performance change in a, let's say, positive way in the placebo group in red, because we have faster reaction times after the procedure compared to before and compared to the 2 control groups. And from a neurophysiological point of view, we were interested, first of all, to replicate the findings of our previous studies on the involvement of the motor system. And for this reason, we concentrate our investigation to two central electrodes, C1 and C2, and we extracted a differential wave, which is a lateralized pre-movement wave before the EMG onset, telling us how much the contralateral motor cortex is involved in controlling the movement. And we found that indeed the procedure worked in inducing a more negative activation in the placebo group after the procedure compared to the session before. Because we can see that this lateralized pre-movement way is more evident in the placebo group compared to the other two groups. Instead, in the control groups, control no tense, we have no types of modulation. In the control tense, we have a negative pre-movement wave before the procedure, which is then zero after the procedure. We are trying now to understand this opposite pattern in the control tense group. Interestingly, this lateralized pre-movement wave correlates with the changes in motor performance, meaning that those participants who have a more lateralized pre-movement wave also have faster reaction times after the procedure than before. This type of study was also planned to allow us to investigate what happens before the go cue and after the warning cue. So in that time window in which participants are anticipating the go cue. As we know from the literature, the placebo effect is due to expectation of a benefit of an effect. And therefore, we were interested in extracting a special wave, which is the contingent negative variation, that is a wave consistently associated to anticipation and can be mostly recorded from a central electrode, which is CZ. So our hypothesis was that this wave should be modulated in the placebo group compared to the other two groups, and this modulation should also correlate with some changes in motor performance. However, we don't find any types of modulation in this wave, not significantly at least, even when dissecting this wave in two components, the early component, which is extracted from 2 seconds until 2.5 seconds before the go cue, and the late CMV, which is instead up to 1 second before the S2. So as you can see here, we found a negative component, late CMV, in all the groups, which was not modulated depending on the intervention. We were also then asking ourselves, could it be that some changes occur before the warning cue? So are we able to extract some cognitive mechanisms that are not related to the movement per se, so not related to motor preparation, but to the fact that in a certain way, the warning cue becomes more, let's say, salient or arousing in some individuals compared to others. And the hypothesis is that maybe the placebo procedure induce this kind of better processing of the warning cue that then in turn allows individual to be faster to react to the go cue. To answer these questions, we move with an hypothesis-driven approach based on the literature, suggesting that there are some brain oscillation like alpha and beta oscillation that could be related to a better processing of visual signals. Well, alpha processing, we know that alpha oscillation are more related to the ability to process in a more accurate and reactive way some visual or sensory signals. Whereas beta oscillations have been related more properly to a more top-down, better processing of a general like arousal or vigilance of the individuals. And to answer this question, we concentrate our investigation on two clusters, a frontal and a parietal cluster, on the right hemisphere that have been consistently associated with attentional modulation. And our preliminary data suggests that indeed the placebo procedure seems to enhance beta power both in the anterior and posterior cluster after the procedure compared to the session before. Instead, in the control tense group, we found an enhancement of beta power only in the posterior cluster, but not in the anterior cluster. No change has been observed in the control no tense group. So now we are trying to, let's say, add some pieces and to put all these pieces of the puzzles together with more sophisticated analysis, allowing us to run some additional or moderation analysis, and especially to try to combine all these clusters, frontal, parietal, and central cluster, with some analysis of connectivity. So to conclude, we can say from these last preliminary results that the placebo effect induces faster movement and greater motor cortex lateralization before movement onset, and this goes in line with previous studies on the motor system. This modulation appears to be favored by a top-down attentional engagement in a frontoparietal network, although this is an hypothesis at the moment, and we are going to enlarge this investigation, as I say, with analysis of connectivity. Of course, now I presented like in different pieces of the puzzle with the behavioral level, with the cognitive and psychological level, with the brain level, but we know that also the body is involved in this, like, I mean here, physiological functions, for instance, electromyographic functions, muscle activity, or respiration, which is very much involved in motor function and performance, but also heartbeat or skin conductance. All these physiological functions of the body can influence and can play a role in this field. And of course, the challenge here is to put all these pieces of the puzzles together in a more holistic framework that allow us to combine the different level of investigation. So I hope that I convince you that studying the placebo effect in the motor domain is an interesting field of research, because it can help at future implication to promote motor functions in patients with motor deficit, to improve motor performance in sports, to disentangle the behavioral and subjective components, and also to uncover eventual general and domain-specific brain circuits that could be involved in the placebo effect in different domains like pain or in the motor domain. So I would like to thank all the people who helped in conducting this research, and you for your attention.